difficulties during your I got the homework from two participants only, and if others participants have faced any difficulties during their analysis, you may ask now before starting today's session. Today we'll start target-based drug designing. So before going to start, if anyone having any doubt or faced any difficulties during the process, please let me know so that I can proceed after the so I have given two script, Python script in the message. Please copy this script and paste it in your any Word file or Notepad file. It will be required during our today's session. So I think nobody has any doubt. So I'm starting today's session. Drug analysis is a big process and it will take at least 3 to 4 classes to complete the gene network analysis. So I am not going to describe the gene network analysis here. So I will start the and select a target from the literature, and if you find that your protein target from your planned work, okay. So if the target is available in the database, there is no problem. You can download, search the target, and it can be downloaded very easily as we downloaded the ligand file. And if it is not available in the database, then we have to predict its quaternary structure, the quaternary structure of the protein to be predicted by the process known as homology modeling. So let us see how to download the protein structure from the database. So as we used PubChem for the retrieval of ligands, but in case of protein, we will use protein data bank. So simply type the protein data bank and you will find RCSB protein, RCSB PDB website. Just click on this website. In the top right corner, you will find one search bar. You can search the name of your protein of interest. Just for an example, I am searching a protein of human epidermal growth factor receptor. So simply I will type the name of the protein or code of the protein. So the code of the protein epidermal growth factor regulator is the EGFR. So simply type the code of your protein. You can type the full name of your protein also and click on the search icon. So this EGFR protein is available not only in the human being but also in other animals or other species of the animals. So if you are working with human disease, you need the protein of isolated from Homo sapiens. So in the left hand side, you can find the scientific name of source organism. So if you click in the more, you will get the different kinds of animals present in the database. So as we need Homo sapiens, I will just tick the Homo sapiens and click on the play icon. So after that, you will get that there are 516 structure available in the protein data bank of EGFR isolated from human being. So which protein you should select for your molecular docking or molecular dynamic simulation or other studies. So first of all, if the methods of uh, isolation of your protein is X-ray diffraction, it will be shown the better result. Another one is the so I will discuss it later on. Just see, this is the 4-letter PDB ID of your protein. So if I select this PDB or protein, the ID I will mention as 1WT5. This is the PDB ID of this protein. So this is written as the crystal structure of a homogenized antibody FB528. So it is not the EGFR. So I will not select this one. So I'll scroll down below and I will try to find. See, this is the crystal structure of EGFR covalently binding with WZ402. This is the ligand binding with the protein. EGFR. So you can get the different types of ligand binding with the same protein. This is the EGFR kinase domain in complex with mutant selective allostatic inhibitor. So let us open this one. So when we click the protein, you will find the details about this protein. So first of all, the organism is Homo sapiens. Then if you scroll down, you will get the methods of isolation is X-ray diffraction. The another point should you be remembered that when you select a protein, the resolution should be very high. So higher the resolution means the structure of the protein is more accurate or perfect. So you can select this structure for your molecular docking analysis. So to download, just click on the download files, go to the PDB format, click on it, and you will be you save this protein in your desired directory. So my protein has been downloaded. This is my protein. Now there is so many software is there to open the protein file. So you can use the Discovery Studio visualizer. You can open the Rasmol. There is so many software. There is Pyrex, Pymol. There is so many software you can use to visualize this protein. So I am having an paid software known as Molecular Virtual Docker. I am opening this protein in this software. So when I right click, double click this software, it will show that this protein is contained two cofactor, 398 molecule of water. There is two chain of the protein and three ligand is binded with the protein. So if I remove the cofactors, water molecules, ligands and click import, then you will get the structure of your protein. So this is the structure of your EGFR protein. So you can work with this protein because it is ready for work. But in case this protein is not available in the database, so what can you do? For this, you have to predict the secondary quaternary structure of your amino acid sequence. So see, there is two chain. There is chain A and chain B. So if I uncheck the chain B, this is the chain A of EGFR protein. And if I uncheck chain A, this is the chain B of the protein. Okay. So like that, we can visualize our protein structure. There is another important software. This is known as Chimera. You can visualize your protein in Chimera also. So this is a process by which we will select our protein for molecular docking or molecular dynamic simulation studies. Whatever structure you will select for your studies, you should mention the PDB ID in your manuscript. Whichever structure you are working with, you should give the PDB ID in the manuscript. Okay. So this is the process by which we can get the 3D structure or quaternary structure of the target molecule or target protein. So if you are working with any bacteria or any other organism, you just type the name of protein and from the left side menu, you just select the name of your species you are working on. Okay. And if it is not available, you have to go for homology modeling. So this is the process by which we can download the protein structure. Now we'll see how to predict the quaternary structure of protein if it is not available in the protein database. So for this, you need some software. One is modeler, second one is chimera, and you need the script I have given earlier. And one important things you require is the amino acid sequence. So the requirement for homology modeling of your protein is two software and one amino acid sequence. First software is modeler, second one is chimera, and third one is amino acid sequence for your protein. So I am showing you from where you can get the software. First of all, you need modeler. For modeler, you just type modeler and open the first link. So this is the official site of modeler. In the left hand side, you will find download and installation. From the download and installation, just click it and scroll down below. So there is showing us there is various installation file for Windows as well as Mac, Linux, and there is 32 bits, 64 bits. So according to your system, you just click it and it will be prompt to download the software. But this software needs the license to activate. When you try to install the software, you need the activation key. For getting the activation key, you have to register it. So below the download and installation, you will find a box known as registration. Just click on it, and when you scroll down, you will get one form. In this form, you have to give your name, your title, name of your institution, address, and most importantly, you need an institutional ID. Institutional email ID is required. Your Gmail, your Yahoo Mail, your Outlook, your other type of email will not accepted by the modeler. Only the institutional ID is required to get the free registration license. So if your institution giving you the institutional email ID, just put your institutional ID, then select your operating system and click on agree and accepted. Within 2 to 3 days, you will get the license key in your registered email ID. So if you are having institutional ID, just go for the registration process. Okay. So without the registration key, you can't install the software. For Chimera, just search UCSF Chimera. So this is the UCSF Chimera. So open the first link. In the left hand side, go to the download option, and according to your system requirement, you can choose the software and click to install. It is free of cost. You can download it easily. Okay. So we need two software. One is modeler, second one is chimera. So after installation, So in the modular website, in the below, you can see the tutorial. In the tutorial section, you will get all the codes. The codes I have provided, it is also available in the tutorial section of the modular. But it is written very complex. So I have modified this script, and you just need to change few descriptors of your script to run the modular. So first of all, if you are having the amino acid sequence, then you will start from the converting your amino acid sequence to .PAIR format. Okay, if you are having the amino acid sequence, then you will start from the converting your sequence to PAIR format. So I will show you how to convert your sequence to PAIR format. As I don't have any sequence, so I will try to get one sequence from the UniProt. UniProt is another database for protein. So here I will type the protein name. So for example, I am typing typing one protein name in UniProt. So see, this is the UniProt ID, that is P07947, and this is the YES. YE is the code for the protein named as human tyrosine protein kinase. Yes, YES. So I will just click on this protein. You will get the details of all the details from the protein, but there is not available the structure. So I am looking for the amino acid sequence from this protein. So in the left hand side, you will see there is a option known as family and domain. Just click on this family and domain, scroll down, and you will see there are some region available in the family and domain. So first one is region 1 to 45. Second one is compositional bias. Third one is domain 1. Second one is domain 2. Third one is domain 3. So see, the fourth, the domain 3 is the protein kinase. So I am interested to work with the protein kinase, the kinase part of the protein, so that I will select the sequence which is starting from 277 to 513. So to get this amino acid sequence, I will just click on the blast. So when I click on the blast, I will get the amino acid sequence. See, this is the amino acid sequence of the protein tyrosine kinase. So this is the amino acid sequence. So I just select the amino acid sequence, right click on it, and click on copy to copy the sequence. Then you just create a folder without having any space. Name it without having any space. So I'm writing this folder as homology modeling. I have not given any space in this name. You should not give any space in this folder. I am opening this folder, right click, and open one notepad. So this is one notepad. I am pasting the sequence here. Now I will save this sequence as FASTA format, because This format of the sequence is known as FASTA sequence. This is the sequence, and the first part of this sequence is written such a way which is known as dot FASTA format. So I am saving this sequence as FASTA format. To save this format as FASTA, just click on File, go to Save As, then give the name of your protein or the ID of your protein, that was P07 P07 947. So I am giving the name P07 947. And after the name, I will put one dot and write FASTA. And save as type, I will click on all files and click on save. So I will close this one. Now you can see in my folder there is a file named as P07947.FASTA file. So the amino acid sequence I have converted into .FASTA format. So again, I will open the notepad, will paste the same sequence here to convert the sequence as a .PIR sequence. Okay. To convert the .PIR sequence, there is a process. I have mentioned it in the chat box. You can see in the last script, there is written as greater than P1 colon 1 YES, then sequence colon 1 ES, then there is 8 colon, then your amino acid sequence, then star. That means the line should start with greater than PI. So I will add greater than symbol, then P1, P should be in capital, P1, then I will colon. After giving the colon, I should give the 4-digit protein ID. So in case of my protein, the ID is 1YES. This is the protein ID of my, I have the selected protein ID is 1YES. Then I will give one, I will click on enter, and second line I will write sequence SEQUENCE. After writing the sequence, I will give one column. After giving one column, I will write the protein. Writing the protein, I will again 1, 2, 3, 4, 5, 6, 7. Giving column. There will be amino acid. I put one. So the PIR protein sequence, you the coordinate structure. So P1, then given one colon and give any 4-digit code of your protein. In the second line, you write sequence, give one colon and write the name again and give 8 colon. After that, you paste your sequence, and after completing the sequence, you give one star, then your sequence will be converted into .PIR format. So from where you can get your sequence, if you're working any, you can for the sequencing, and from the sequencing you can get the idea of MRP. If you are you can so then the sequences. So if you are having yes, you can you start your homology. If you are not having this, you can start in the so maybe your protein is not able to PDA, then no able the is able in your uniprot or not. The is able in the uniprot, you can easily design your protein cotton structure. And if it is not in the uniprot, you have to sequence it and get analysis. So after formatting a sequence in PA, I say and earlier we have written as dot fasta, but now I will written as dot PIR because this is the PIR format of sequence. Then select all files and click save. So I'll close this notepad, and if I go to the folder, you can see there is now three file. One is notepad. I will delete it. So there is two file. One is the FASTA format of the and PIR format of sequence. So if you click on the last, then you will get the format. It is known as FASTA format. But if you want to convert your sequence to the FASTA format, you just give a greater than sign, then write SP, then one dash, and write your name, and you can give the number of the starting sequence to ending sequence. As this is started as 277 to 530, it is written as 270 to 530. But if in your case you are working with a emergency sequence you generated from the sequencing, then you no need to give the sequence range. Just give one greater than simple and write SP, then protein ID, and paste your amino acid sequence. It will be treated as a FASTA format. Or you can Google the conversion of amino acid sequence to FASTA format. There will be there will so many tool you will got where you if you paste your amino acid sequence and click, it will convert your sequence to FASTA format. Like that, if you paste your sequence here and the output format is FASTA and click submit, your format will be automatically convert into FASTA. But you not need to you not need any tool to convert the format because you have sequence just in the first line you give one greater than sign, write SP, give one dot and give your protein name. If anyone having any doubt, please let me know because this is very important process which is conversion of amino acid sequence into .pa format and .fasta format. If there is any mistake, the when I run the modeler, it will not work. There will show an error. So it is very important. Clear to everyone. Yes, the format converter can convert a sequence to a PI format, but I have seen that when we use the converter to convert a sequence to PIR, it is not uh, detected by the modeler. Okay, so when we convert the sequence with a sequence converter, then when we put it in the modeler, it is show error. So I recommend to format the sequence into PIR manually, and it is not very difficult to convert the problem to convert the sequence. So Abhishek, which part you want to repeat? Okay, for the first part, when you search your protein in UniProt and click on familiar domain, you will get the different domains available there. Okay, so click on the blast of your desired domain. As I am working on protein kinase, so I will click on the blast for the protein kinase. When I click on the blast, the FASTA format of the sequence of this protein will be shown. So this is the FASTA format. I copy this and paste in Notepad and save as a FASTA. It will treat as a FASTA. But if you are having the sequence of your own word, then how you can convert this file into FASTA format? So for this, open the Notepad, paste your sequence in the Notepad. In the first line, you write greater than symbol, then write SP, then give one dash, then write your protein name and save this file as .fasta format. Then it will be treated as a FASTA file of your sequence. I hope you have understood now. Okay. So now we have our protein sequence of protein sequence of interest we have, and we have converted it into .fasta format and .pa format of our sequence. Now we have to design the quaternary structure of this protein. So modeler will design the structure for us. But how it will design? Because it needs a template. So if you want to make an idol and you have gone and ordered to make a idol of any god. So they will prepare accordingly because they have the idea how this god is looking. So you are going to any shop and you have ordered to prepare one uh, idol of god Ganesh. So he will prepare the structure of Ganesh as he know how Ganesh is look. But if you go here and say to him that please make a idol of my friend, so he can't make the idol of your friend because he don't know how your friend is look. So you have to give a photo of your friend so that he can see the photo and can prepare the idol of your friend. So you have to give a basic idea that how your friend is look. So in this case, in the modeler, we have to give one structure, similar structure of your sequence so that modeler can see, okay, this sequence is folded like this. So the unknown sequence will be folded like this. So I have to give one template structure or template protein structure to the modeler similar to the sequence of my interest so that modeler can understand how it will design or how it is um, fold the amino acid to make a tertiary structure. So for that, we have to blast our protein sequence so that we can get a similar structure available in the database. Okay. So for that, we will perform a protein blast. So what we'll do in protein blast? In protein blast, we will upload our FASTA format of our sequence. Then we'll do blast, and it will give the result of identical structure of our sequence. So higher the identical the template structure, the more accurate quarterly structure will be predicted by the modeler software. So basically, it is considered that the similarity should be more than 40%. The template structure and your query structure similarity should be higher than 40%. But it is better if the similarity is more than 90%, more than 95%, it will give more accurate structure of your query sequence. But it is also considerable if it is uh, greater than the 40%. But the probability or the accuracy of the structure will not be so high if you consider the identity is in low value. So how we'll get this? For this, we have to visit one website. Everyone know this one. This is NCBI. So in NCBI, we directly NCBI blast. So this is the NCBI blast. We open it. So there is different kind of blast is available in this portal. One is uh, nucleotide blast, one is B blast, one is T blast, one is protein blast. So we are giving the protein sequence and we need the protein sequence in back. So we'll select the protein blast. So just click on the protein blast. So after selecting the protein blast, you have to select the FASTA sequence here. Now click the choose file, go to the folder and select the FASTA file you have saved. So I have given the FASTA file of my sequence. Then I need a protein structure. So where the protein structure is available? Protein structure is available in protein data bank. So as it is available in the protein data bank, I will select the database as PDB. It is protein data bank. The database where it will be searched, that is protein data bank. As we need the protein structure. Then we will scroll down and click on blast. It will take few minutes generate the data. So see, they have generated so many. There is 100 sequence they have generated similar to my query sequence. So see, there is written as percent identical. The percent identical is first one is 89.76%, second one is 89.37%. So this is very good score. So we can check any of this protein structure. But before going to the getting the structure, we have to check the quality of the protein structure. Like that, if you are giving a photo to the to make a idol, and you give a photo which where the face part is uh, teared, or there is blurry face is in this photo, then it is difficult for him to design the idol for your given photos. Okay, so I have to see, is this protein, that quality of this protein is good or not. To see this, you have to open the software Chimera. So this is the chimera. Now, see, we are working with a protein of Homo sapiens, but the ideas, the percent identity is matched with the galas. So it is not necessarily we have to choose the template from the same organism. We can choose the template from any organism, but the percent identity should be high and the quality of the protein should be good. So it is not necessary that we are working on Homo sapiens, so we have to select template from the Homo sapiens. We can choose the protein structure for any organism. So first of all, we'll see the protein quality of Homo sapiens. Okay. So what is the protein ID? That is HJF3. So I will open the chimera, go to file and get fetch by ID. So this is the PDB ID given there, that is 8JF3. I will write the protein ID here, 8JF3. And click fetch. Type the protein ID here. The protein ID is 8JF3. 8JF3. This is the protein structure similar to the query sequence. I will download this protein. So I no need to download because it is already fetched. So see, if I look at the structure, which is a template structure from the Homo sapiens, you can see there is some dotted line in between the protein structure. There is some dotted line. See, there is some dotted line is there. And there is some dotted line. This dotted line means in this protein structure, the in the middle of the sequence, these sequence are missing. There was no information of the sequence, or when it is crystalline, this part of the sequence of this part was destroyed. So this is not a complete protein structure because the some part of the protein is missing here. So as the protein structure is not complete and some part of this protein is part of the amino acid sequence is missing, so we can't take this sample as a template because it will not give us the accurate structure. So we will not consider this structure. So if we consider the another protein which is 3U4W, go to chimera again, go to file, fetch by ID 3U4W and click fetch. See, in below the ID is fetched. See, in this structure, in this blue structure, the structure is complete. There is no dotted line. From the starting to ending, there is no dotted line. That means this structure is complete and it is a very good quality structure. So we can choose this structure for our template. So in spite of having a template from Homo sapiens or the same organism, we are selecting a structure from another organism which is Gallus gallus. So we will download this protein structure. How can we download? Again, go to the PDB, give the PDB ID, click on the download file and download in PDB format and save it in your folder you have prepared. So I will close the Chimera. So now in this folder there is three files. One is .py file, a 5-star file. One is the protein template file. Okay. So up to this, if you are having any problem, please let me know because it is a little bit complex process. So if you are having any difficulties, please. On which part should you want to repeat? Okay. So if you having any difficulties or confusion, please let me know because it is very complex process. Okay. Repeat the last part in Chimera dotted line. Okay. So when I open the Chimera, so as I already okay. So from the file, you can fetch the ID. Okay. Give the ID here, and you can fetch the structure of your protein. So the ID was 8JF3 and click on fetch. It will fetch the structure of your protein. So when the structure of your protein will get, see, when you see the structure of your protein, you can see in the protein, there are some dotted line in between. See, there is some dotted line. There is some dotted line. There is some dotted line. See, this part is broken and this part is also broken. And there is some dotted line. That means that in this structure, the amino acid sequence for this part is missing or damaged. So this structure is not complete. In this structure, in various region, it has been broken down and the part of the amino acid sequence is missing. So this type of amino protein structure we can't consider for homology modeling or take as a template for homology modeling. See, this is totally broken. This part is broken and that part is broken. This dotted line means there is no amino acid sequence present there. So that means in the protein sequence, it should be a linear part and folded in such a way to that it create a quaternary structure. But if it is broken in between, there will be a lack of amino acid sequence. So we can't take those protein as a template. See, in various places, it is broken and the sequence of amino acid is missing. See, that is the broken part. Clear? Okay. Now go to the insert message and you file to script. So if you have copied this script in Word, or in Notepad, just open it. I am opening it in Word to show you. Just a second. Okay. So there is two Python script I have provided. So from the first script, this is the script. The highlighted area is the script. So you have to copy this script and open a notepad. Copy this script and paste it on notepad. Now see, there is some yellow marked portion is this script. So first of all, this 3U4W is highlighted. What is this 3U4W? If you open the folder, you will get the protein structure we have downloaded. It is known as 3U4W. Okay. So in this, in your case, the template structure name should be written here. In this script, you should write the protein name of your template protein structure. In my case, it is 3U4W, so I have written 3U4W. Okay. Secondly, there is written as first A and last A. If we go to the NCBI blast, you can see that after the protein ID, there is written as A. That means this sequence of our query sequence is similar to the A chain of the protein. You all know that there may be more than one chain in a protein structure. Chain A, chain B, chain C, chain C. So in my case, chain A is similar to my query sequence. So I have written as chain A. But in case of this, if your structure having more than one chain, then the first one will be chain A and last one will be the number of chain. If there is two chain in the protein, it will be first A, second B, last B. If there is three chain, then first one will be A and last one is C. Like that, there will be first to last chain number. Then in the below line, you can see there is another word is highlighted. There is written as 3U4W. That means this is the protein name. You change this name as per your template structure. After that, there is written as A. As I mentioned, this is the chain which is similar to my query sequence. If I chose this protein, if I chose this protein that is 3D7U, it is written that the chain B is similar to my query sequence. So if I chose 73U, then I will write 73U B, because in that protein, B chain is similar to my query sequence. Okay. Then it is written as 3U4W.pdb. If I open the folder and see the full name of my protein, see, The full name of my protein is 3U4W.pdb. The full name of this protein is 3U4W.pdb. PDB is the file format. So I have to write the full name of full file name. So in your case, that will be your protein name and followed by .pdb. Now, most important part is the next line, which is written as .pir. So as I written the query template now name is P07947.pir, the name should be the same as written in the query PIR format. So the name given in the PIR file, the name should be same as written here. After that, you have to give the 4-digit protein name. As we written the protein name as 1YES, so we should give the same name here. Okay. So this was the first PIR format, sorry, Python format for the first script. So what you should replace here, first one is the protein name, protein chain name with name, protein name, full protein name, PIR file name, and the protein name given in the PIR file. After replace all the replacement, just go to the file, go to the save as, and write as one underscore alignment. As it is the script for alignment. Then click give one dot and write POI. POI means Python script is written as .py and save as all files and click save. So first format, first Python script was been written. Now the second script, just copy the script, paste it the notepad. In second script, you just repeat the written what you have written in the first script here, just copy and paste there. Then write the protein name as written here. And the ending model is the process. We are creating 10 model for the same amino acid sequence. There will be 10 protein model. From the 10 model, we will select which one the best protein structure for our further work. You can select 5 models, you can select 10, 20 models, you can select 50 models. How many model you want to generate by the modeler, so that you can shortlist it from them the best model you can write here. So in this case, I am selecting 10 model, so that from the 10 model, I will select the best model for my further work. So I have written 10. So after replacing all the things, go to the file again, go to the save as, write as 2 underscore, then write anything like model dot py. All files. Save. So 2 Python script is ready for our work. So now again, if you are having any doubt, please let me know. Till now, if you are having any doubt, please let me know before proceed the further process. Okay, so I think there is no doubt. So now we'll open the modeler. Click this is the command prompt of modeler software. So first of all, we have to select our folder directory. So how we can select our folder directory? We have to write CD. We have to write the CD, then give the space. When you open your folder and click on the address bar, you will get the folder path. So just copy the path, go to the modeler and paste it and click enter. So we have entered in our working folder. Now we have to give command to modeler to run the Python script. For that, the different version of modeler have the different version of command prompt. For my case, the command prompt for modeler is MOD 10.5. So different version of modeler have different command prompt. It will be written here. So in my case, the command prompt is modeler 10.5. So I will write MOD 10.5, give one space, and I will write one and will give one tab. Why I give write one? Because the first script I have written as one underscore alignment. So when we type the first letter of the file and click on tab, it will automatically detect the file present in the folder. So I have write one because my file name is starting with one, and I have entered the tab button so that it detect the Python file of my interest. And I will click enter. It will take few times. After the running, you will get the first alignment result. So the run is complete. After completing the run, it will show again like that. So if I go to the folder, you can see there was 5 file earlier, but now there is 7 files, and 2-3 files are added newly. So I will type the second script. Again, I will type mod 10.5, give one space, give two, and tab, because my second script is written as two underscore model. So I will give enter, and it will take few minutes to complete the modeling process. So wait for few minutes until the modeler complete the modeling the structure of your query and amino acid sequence. After the completing the run, it will show the same line as appeared earlier. So wait for few minutes. So the process has been completed. We will close the modeler. So when we open the file folder, you can see there are 10 protein structure has been created by the modeler. This is 1YES, as we named the protein as 1YES, and the last digit 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. As we mentioned that it should create 10 protein structure, so it has created 10 protein structure. If we selected 5, then it will create 5. If we create set 20, then it will be created 20 protein structure. So which protein structure should we select for our further work? So for that, there will be a file named as 2 underscore model building. When we open this log file and just scroll down the end part of this report. So this is the end part of the report, and you can get the 10 protein ID generated with the modeler is written here. And there is some score written as DOPE score. DOPE score means discrete optimized protein energy. And lower the DOPE score is higher the or better the structure. So in this case, you can see in everywhere in 1, 2, 3, 2, 2, you can see this protein structure is showing the lowest DOPE score. That is 31,772 is the lowest DOPE score shown by the protein ID 7. So the 7 protein created by the modeler shown the lowest DOPE score. And we know the lower the DOPE score, the better or the accurate the structure. So we'll select the 7th protein structure for our further analysis. So to see the quality of our protein, we'll check it in the Ramachandran plot. Ramachandran plot will open one website known as saves.mbi.ucla.edu. So this is the server. I will choose the file. There is a choose file. I will choose the file from this folder. This seventh protein structure I will open here and click run program. After that, in project, click start. So the result has been generated. Click on result. The left hand side, click on the Ramachandran plot. So this is the Ramachandran plot of our predicted protein structure. So this is the Ramachandran plot. If you scroll down, you will find 94.5% residues in most favored region, 5% residues are in additional allowed region, 0.5% residues in generously allowed region, and 0% residues in disallowed region. And if you scroll down, you will find that a good quality model would be expected to have over 90% in the most favored region. So, to be a good protein, the most favored region should be 90% or above. But in our case, it is 94.5%. So, definitely we can consider that our protein structure is quality is very good for further analysis. So, this is the Ramachandran plot of your protein. You can download the structure by clicking the PDF and it will save in PDF format. So you can use it for your manuscript or in your thesis. So by this process, we can analyze the quality of our protein. So I found that modeler have prepared a very good quality of protein of our query sequence. Okay, so this is the result of Ramachandran plot. So by this, we can prepare the quaternary structure of protein from an unknown amino acid sequence. So this was the process. This is the definitely low quality. Yes, high similarity is required for protein modeling. And if it is high, similarity is high, the query coverage will be high. Okay. If the query similarity is high, then definitely the query coverage will be high. But in some cases, coverage will be high, but similarity will be low. In that case, we can't select this because we have to select high similarity above 40%. Okay. So with the low similarity, there may be a template with high query coverage because it may cover the sequences from different part of the template sequence, but we need high similarity sequence rather than the high quality query coverage. Okay. So if anyone having any problem or doubt, please ask me. Yes, there is another tool. I will discuss it tomorrow. I will show the same process with another tool tomorrow because but this the tool I will discuss tomorrow is very easy, but the accuracy of prediction of the protein sequence is very high in modeler, and it is widely accepted process or modeling process of the protein. So it is better, or I will recommend to use the modeler for protein modeling. But tomorrow I will show the Swiss modeler server to predict the protein model. But I will recommend modeler because it is having the high accuracy than the Swiss modeler. So I will show you the process by which you can create a protein structure using Swiss modeler. It will take 1/4 time taken by modeler to create a protein model. Tomorrow I will discuss about it, and tomorrow I will start the molecular docking process. So if you are having institution ID, I will suggest you please go for the registration process. It is free, and get the license key and save the script I have given and try it yourself. So I think there is no doubt. Anyone don't having any doubt or confusion? So thank you everyone and good night. We'll see you tomorrow again.
Thank you, Rajat, for this nice session, and thank you to the participants. So, okay, we will see you in next session. Thank you.